Okay, I'm going to make the executive decision and start. So welcome everyone. Feeding Willow to Livestock Network event. It's great to have the IF Field Lab team here to talk us through what they've been up to and their results. I'm John Haines. I'm the Agroforestry Advisor at the Soil Association. So work with farmers across the UK, all types of farmers, worth saying, to support them to integrate trees into their farming systems. It's great to be here. Great to have such a big attendee list at the moment. So more people coming in, but that's great. So a couple of housekeeping rules from me. Rules, that sounds a bit scary, but so the session is being recorded. You'll notice as you've come in, you're automatically muted and webcams are disabled at the moment. We are hoping to turn on or allow people to turn on their webcams if they'd like during the Q&A session that we will have after the speakers have said their piece. And then the only other element was if you have questions. So I think we're fairly tight on time. Maybe we're covering quite a large topic, and there's lots of enthusiasm from all different angles. So we'll I say me, but Tabitha hopefully will try and keep us on time as much as possible. And in that vein, if all questions could try and go in the Q&A box at the top, you can see fourth icon along the top of your screens. If we could put any questions you have, we've got 10 minutes allocated to questions. We might be able to have a bit longer if we can keep everyone to time. And then there's also on that Q&A section, for those that don't know, there's an upvote ability to upvote questions. So if there's a question that you see that is really interesting, or you're dying to know the answer to, by all means upvote, and that will give me as Q&A, as person reading out the questions in time, the idea that it's an important one to prioritize. So thank you very much. I'll pop that in the chat for everyone that perhaps joins late as well. But hopefully it's going to be a fantastic webinar and some really great speakers and what is a great trial. So without further ado, I'm going to hand over to Tabitha, who's going to introduce Innovative Farmers and the trial that they've been up to. Thanks, Tabitha.
Thanks, John. Hello, everybody. For those of you that I haven't met before, I'm one of the farming advisors within the farming and land use team. So similar role to John, but very general. So I don't specialize in trees. I seem to have specialized in sheep for my sins. And I also farm with my husband up on Hadrian's Wall in Northumberland as well, which I think is slightly how I've got dragged into the sheep focus. So today I'm just going to introduce you to Innovative Farmers and our Willow project that we've been doing. I know some of you will know about it, but we just wanted to make sure everybody was on the same page before we get to the exciting stuff of what we found. So those of you that haven't come across Innovative Farmers, it is a network of farmers and growers, and it's all about on-farm research, and it's all farmer-led. So you come to us with ideas. We've got relatively small, in the grand schemes of research, pots of money, but enough to make sure that you get a researcher and we get some good tests done and get some good information back from some on-farm trials. The main way we do this is through something that we refer to as a field lab, because we're doing research on farm. That's where that's all come from. And the field lab is kind of culmination of the on-farm research. You get dedicated support from a coordinator and a researcher. So in terms of this Willow project, I've been the coordinator on behalf of IF, but from a Soil Association point of view. And we've got Nigel Kendall here from Nottingham University, who's been our researcher. The farm we've been using is Tom Fairfax from Mindrum, and you're going to hear from him in a few minutes' time about the kind of farm learnings in terms of that, whereas Nigel's going to talk about the research learnings. And we get really good media pickup for field labs. I think they have a good feel about them because everybody's very enthusiastic, and that's how they've come about. The media, particularly farming media and stuff, really like picking them up. So lots of things happen with that. You might have heard about this project on Farming Today at the beginning of the year. And I think Tom was speaking to the Farmers Guardian last week about it as well. So you'll see a bit more of it coming up like that. We also do lots of lovely knowledge exchange events. We did the one for this project back in September, which we used as the last point in the project where we were collecting blood samples. So Nigel was up in Northumberland with us as well, and we could get everything together in a couple of days. So we did it kind of before the end of the project in this situation, but it was a good use of everybody's time. So in terms of how they look across the kind of the whole project, we've done well over 150 field labs now, which has covered 750 plus trialists, and we've worked with over 40 research organisations, and all of those numbers are going up almost on a daily basis. So those figures, that's why the pluses are there, because it's very hard to keep those figures up to date. But it's worth having a little pop on the website and seeing what has been looked at. But also on the website, you can fill in an expression of interest if there's anything that you particularly want to look at. If you've already got a group of farmers together, by all means, come to us as a group. If it's an idea that you haven't spoken to any other farmers about, or you don't know which farmers to approach about it, then we're very happy to kind of get people together and get a group together as well. Sorry, I just noticed in the chat, if people can't see the slides, logging out and logging back on again tends to work. And then you can also do the full restart if that doesn't work. This does tend to happen for one or two people, unfortunately. We'll also share the slides around at the end, so don't panic too much. So the field lab official title was Evaluating the Effect of Feeding Willow Leaves on Growth Rates in Weaned Lambs. And you can see the project aims there. So there was this work had started originally. So Nigel has done work at Nottingham in a very lab-based setting, and they know that there is a blood response from Willow in terms of cobalt. What we really wanted to look at in terms of this project is whether we saw that expected production response that we would also see if we supplemented cobalt in another way. So that's what we were really looking at. We did do blood tests and things as well, just to confirm that we were getting that blood response and to support anything that we did find. But that was the kind of the aim, was very much about this. Although in the test setting, we weren't able to do it fully in a practical way, how we would recommend it on farm, but it was moving towards that. How would this look on farm, and do we get the production response? So in terms of the trial design, we started at weaning. So it was day one. Sam the shepherd had weaned 2 groups of lambs for us, and we set away starting the project on the same day. It was a very long day for everybody involved. We ended up, through enthusiasm from Sam and Nigel. I'm blaming Nigel these days, but everybody has a different person to blame. We did 580 just over 580 lambs, and they were all weighed, and then we split them into matched groups. They were from 2 different groups of ewes, so we had to kind of use that information as well to make sure that the groups were matched. They were then going on to a lovely herbal ley that was quite rich in red clover that Tom had next to the sheep pens here. And they were rotating around that. And then we weighed them weekly, and then the test group got access to the Willow on that weekly basis as well. We did bloods at the start point. So we did bloods on I think it was the 25th of July, and then about the 25th of August. And then the final bloods were done on maybe the 25th or 26th of September. So we're obviously about to discuss the results in a bit more detail, but we did find there was the kind of maybe the most benefit from this project in terms of stuff that we can take away now is all about on-farm learning, which Tom is going to go through in a bit more detail. But before we get to the farm stuff, we're going to hand over to Nigel, who was going to talk you through the science. So Nigel, are you going to take control of... Oh, you have. There we go.
Let's say I'll put the camera and the mic on, then you might be able to hear me. So which will help. So now I've got 12. So we're going to go through some of the results. The big sort of headline one, if we go straight to that, growth rates, our controller Willow, we had no differences at all. Now looking at these, one of the surprising hard things to find is what is the UK average sheep growth rate. I've had some interesting chats with Tabitha and some other sheep experts that I know in terms of that. We can find a figure of 300, which is what the it's not Eblex anymore, is it? It's the get me acronyms.
AHDB.
AHDB. I was getting APHA in my mind. AHDB beef and lamb put us there, set up target for 300 grams per day. Well, we're nowhere near 300 on these lambs. So, and there was some cracking pasture in front of them. That red clover mix was really looked a lovely pasture. So we might have been expecting these to grow a bit faster. So if we look in there, we've got the selenium cobalt for these. I've just got this because I'll come back to it. But looking at the cobalt and the headlines of cobalt, the cobalt was not quite as low as expected. So there's less of a cobalt gap we needed to supplement. However, it was still at a level that it should have responded. A lower growth rates in the lambs have a lower cobalt or vitamin B12 requirement. This is because it's the vitamin B12 requirement is very limited to the growth rate of the animals. So you actually had a lower requirement. So hence we didn't possibly get the differences between the animals, but something was potentially restricting our growth rate. The willow we're feeding was a little bit lower in cobalt than I expected, and also didn't seem to be quite as palatable, particularly as we got later in the season than previous willow I'd worked with. I think that's probably somewhere being a little bit on right up north on the border. It may have been a bit of a harsher environment than some of the Leicestershire willow I've played with before. Cobalt status, so our vitamin B12s, okay, were there. So there should be two lines down around here somewhere, which 400, above 400 is completely normal, adequate. Between 188 and sort of 400 is, it should be fine, but a little bit more would be better. And there's this low. So you can see at the start of the trial, the lambs came in with a low cobalt status, a low vitamin B12 status, but then we're above this. Their B12 was adequate. There would have been less requirements, so any B12 they got would have gone into the blood rather than being utilised. So and the plasma cobalt, which is sometimes used as a proxy as well, was showing how much cobalt was going in, which quite interestingly, it was only the third blood sample where we had significant amounts of plasma cobalt going in. So we were getting more cobalt intake around that time. So there was no real apparent effect of feeding the willow on the cobalt status, and we'll come into a reason why later on. One of the reasons, well, later on, that's now, I thought this was later, it's now, the intakes were a major issue in terms of willow supplementation. Okay, we fed it because the way we wanted to set the trial up, in the fact we wanted them to graze the same field that they came in. While we had them in for weigh-in, we split them into two groups, offered one group willow, gave one group willow, let them eat it until they were satiated, and then were all let back out into the field. Okay, so from the very initial one where they were actually in, it was quite late at night. I think it was 10 o'clock when we finished, and we decided we'd leave the sheep in on the willow and turn them out in the morning. They ate a lot more. They had a lot more time. So they were eating about 76 grams a head in that time because they overnighted in that sheepfold. However, on the subsequent times where they were in at the willow for about 2 to 3 hours, admittedly the lambs had stopped eating the willow. They ate the willow and stopped, and there was still a bit of willow left. It was weighed in and weighed out, so we know how much we got. The intakes of willow were much lower. And we were only feeding weekly. We were weighing weekly, so this was only going in weekly. So it's a small amount of willow going in weekly. Okay, so these intakes were not actually when you actually did the calculation, didn't cover that supplementation gap. So one reason we didn't get any difference between between the animals. When we fed willow previously, we've been feeding a lot of willow. One of the trials we were feeding 300 grams per day per head, which is quite a lot of willow. So that's about 10% of the dry matter intake. I think if we can get down to 1 to 2% of the dry matter intake, but we were well less than 1% on this. So we didn't get enough willow in, and some of that unfortunately was probably caused by the experimental setup. So I'm now going to have a quick look at selenium. The selenium was low in the pasture, and whether this actually also limited the growth rate. We were well below the requirements. Okay, there. So we're really below the requirements. So we potentially had selenium deficient or selenium marginal lambs, and this might be. It was very interesting when you looked at the individual growth rates. There were some lambs that grew really well, and some that didn't grow hardly at all. So yeah, we had that spread of lamb growth there that was very, very interesting pattern to look at, which may also go back to the bit that some lambs were actually genetically a bit better at coping with the lower selenium status, because all of these things are going to have natural variation. When we looked in the bloods on the selenium status and our glutathione peroxidase, we want to really be which is the functional selenium enzyme, we really want the bloods between these top two. So the bloods weren't in this optimum level up there. You can see they're going down through this suboptimal level into this marginal and deficient level across the trial. And this is similar for plasma selenium. Interestingly, by the September bloods, they'd started to look like they were just turning to come up, and you'd expect the glutathione peroxidase to follow up in due course. So the blood samples confirmed the low selenium status. Okay, we'd sort of had an idea at the middle, but thought, hang on, do we then supplement with selenium? Thought then we're not actually fully addressing the question of what the willow does. You're then adding an extra thing in the middle of a trial, which isn't good. So we kept with the original protocol. Okay, so the low level of selenium potentially may have compromised our growth rates and meant that we were not able to express any cobalt willow effect. Incidentally as well, just another one there, the iodine, plasma ligand iodine was also, we got a marginal level, an adequate level, was also quite low across the board. So our iodine status wasn't brilliant either, which may or may not have affected the growth rates. So the sciencey conclusion is we had low growth rates, probably caused by other confounding mineral deficiencies. The willow leaf intakes were compromised by our experimental setup. And in the future, it would be good to test for and eliminate any of the confounding mineral issues. So if we're doing this again, we'd want to really get a background mineral status. We weren't expecting some of those coming in here, but we were in a completely different field. And look, use a different willow delivery strategy where ideally where we've probably got willow on offer all of the time. But that in an experimental setup means you're probably working with different paddocks or having to try and split lambs in the same paddocks. And to be honest, electric fences don't often work for splitting lambs in the same paddock. Also, whether different varieties or different have different palatability, different I don't really know on that. I know that different varieties have slightly different cobalt content. We've done some work. I've done some work with some of the short rotation coppice, and there's certainly one of the short rotation coppice variety that is higher than any of the natural ones I've seen in cobalt. There's one of those that from a bit of pilot work is very high, and that I believe is me done, and somebody else needs to take control.
Yes, I'll take back control. Thank you, Nigel. And I didn't even have to give you a time warning. I was waiting. We are, just to let everybody know, I am aware that we're zooming through it a little bit, but there will be plenty of times for questions. And the report is we're just editing and stuff at the moment on the final reports that will be coming out soon. But do have a think of your questions, pop them in the Q&A, and we'll come to them after Tom's done his little bit now. Over to you, Tom.
Tremendous, thank you. Would you click through? That's good. Good evening or good afternoon, everybody. What I'm going to do is have a quick chat about the operational outcomes on farm. Just to give you a little bit of context, what we're doing and how we're doing it. What we're here to do is to produce healthy, nutritious food by harnessing the natural productive power of the farm. We're organic and in a regenerative model. So we're really thinking about how we use and maintain the productive power. We're about 4 years into a sort of regeneration phase since we've been farming like this. And our current main effort lies with the profitable regeneration of the ground and the assets. So that basically means we've got to be profitable. But everything else is going back into rebuilding the productive power of the ground. And that's one of the things that we're doing here, is looking at how we generate how we enable the ground to help us deliver business better. I'm using a set of guiding principles. There's lots of amazing doctrinal approaches, all of which are principle driven and all of which are useful. This is my overarching set of principles that I use. And these are what I'm going to just use as a framework to discuss the findings and the outcomes from my end. Just give you a little bit of context where we are. This particular phase of the trial was the third phase for me. In a couple of years ago, we noticed that the lambs, once they're weaned, were actively using willow in the boundaries when it was available, so in hedges, and mainly goat willow. And we noticed that they were actively going to it on a fairly regular basis. They were using it, but they weren't eating it just because it was there. And anybody I did a bit of testing on myself actually, and made some willow tea. I don't know if anybody's bothered with it, but goat willow is pretty bitter, and that may have something to do with it. But so we noticed that this was going on. We did a little bit of punter science in 2023, and I got the vet to take some blood samples from some sheep and put half of them into a field that had willow in it and half of them into a field that didn't. And we doubled in 3 weeks. We doubled the vitamin B12 in the group that had access to willow. Obviously not terribly scientific, but on farm useful. So that's why I rang up Tabitha and we went on to this phase. Now, the farm outcomes, effectively, a great deal. It was extraordinary from my point of view in terms of the information we drew from this, building on information from both within and outside the core project. Firstly, in terms of the context, what was really clear, Nigel was really interesting about this, the impact of the conditions on the way that nutrition happens on farm. Things like willow, like weather, the soil and the ground. If you're like we are in a transitional phase, the ground is changing all the time and becoming more functional from a nutrient point of view. And you can see that. The growing conditions, not only between years, but through years. I don't know whether anybody keeps rain data. I do. And I was looking at my rain graph last year versus the year before. And frankly, we could have been in different continents or hemispheres, let alone different years. So things are changing. And also the farming model that we're in. We're very much attempting to strike a nutrient cycling model. And so again, these are all going to have impacts on the way we manage nutrients. The information and the indicators, for me, it's all about looking at the information, finding out where we are, what's informing the decisions that I'm having to take on a day-to-day basis, but also on a strategic basis. So a lot of looking at the soil. One of the things that was interesting is because we were doing an adaptive grazing model in order to try and keep the sheep in the trial on a consistent grazing as possible. So we actually wasn't quite adaptive, where it was semi-adaptive grazing. It was sort of fast rotational grazing on this lay, it meant that the clover forb lay was getting a lot more rest between being used, and sticking the spade in the ground. The red clover roots were about a third again longer than they would normally be, which of course would make sense for those who are adaptive grazing. So it may well be that there's things going on there in terms of nutrient mining and things like that, which could well be bringing in nutrients from elsewhere. One of the things we looked at, we were looking at bloods and we were looking at weight. Of course, for those of us who manage sheep, the key thing before when you're actually drawing sheep is the feel on the back. But there's other things like the brightness. The thing that the shepherd really noticed was the fact that the difference between the sheep who are getting the cobalt and aren't are the ones. It's a brightness of the wool on the sheep. So there's things like that. We were looking at fecal egg counts, but not. We should have actually probably done this through the trial, but we started looking at it later on. And also things like weed reading, looking at what's growing in the field. So there's a lot of indicators that might be suggesting what we're doing. Curiously, I know that the growth rates were technically low. In fact, as it turned out, I was quite happy with them, and I think probably average rates may or may not be working here. We were grading pretty well when we were drawing the sheep, and in fact, the shepherd got an award halfway through the trial for sheep in spec.
I was going to say incredibly well, grading them incredibly well.
But so from that point of view, I think we've got to be a little bit careful. I was quite pleased with the way that they were growing, and also the way that they were filling, because as I say, it's not just about weight; it's about the hand on the back, which is the most important thing. So looking at the productive power, and this is the model that we're using. It's looking at in this case, a lot of the, if you like, the physical components that actually we use to deliver what we do. It was pretty clear here that actually, if we're going to be at farm scale, if Willow is going to be effective, they have to have access to it on a continuous basis. And my feel very much watching them was where possible, it needs to be accessible in or on the edge of the field. So we're doing quite a lot of agroforestry on that, and we're planting. We're planting a lot of willow and as other things in fields in various configurations, and as an outcome for that, we're very much ramping that forward, which is really exciting, and obviously it has many benefits. The other thing I think was interesting, looking at the forage analysis, was the forage diversity is important, not just for the cobalt, but for all the other things. I'm looking for something that produces selenium, which I think Brazil nuts do, but I don't think they grow in Northumberland. So if anybody has any ideas, let me know.
Ragwort, Tom, ragwort.
Ragwort, yeah. Yeah, my sheep are rather usefully dealing with the ragwort rosettes at the moment, actually. Yeah. But that's useful. But it's also the impact of the management on what we're doing. Very clearly, this field that they're in, we'd already taken a cut of silage off it, and this grazing strategy and the way we were managing it very much. I know it was a grower year, so it's hard to say just in 1 year, but there's clearly the way that we manage things is going to be something that we on farm can look at and use. Also, I thought Nigel's point on the selenium thing was really, really powerful and really useful. This thing about genetics, we just started, we've always had a closed herd of cattle, but we've now gone into a closed flock, and we're breeding our own replacement ewes. And the key thing there is we're really now looking at the choosing the ones that do best on our ground. And I think that's probably quite an important point. And that sort of brings us into the next bit, which is the monitoring. What do we look at? And I apologize for the ChatGPT pictures here, especially you'll see the one of Nigel with his syringe. Clearly, forage analysis is important. What is quite clear in the sort of nutrient cycling model that we're in is that you can't necessarily draw a straight line between what you're putting onto the ground and what is coming out of it, because we're finding quite often that both the livestock and the crops are getting more out of the ground than we might explicitly expect from soil samples and things like that. So we're looking at a lot of levels. We're looking at forage analysis. We're doing metabolic bloods. In fact, my local vet is coming to do the use. I think a lot of us will probably do this for lambing, to actually look at how the animals are actually doing in the current set of conditions. Clearly, we look a lot at soil analysis, not just broad spectrum and soluble nutrient analysis, but also looking at total nutrients, which have become really important in the regenerative model we're in. And also, obviously, performance.
Sorry, Tom, I'm just giving you a 2-minute warning.
Thank you. That's very elegant and actually fits rather well. And finally, it's the integration piece. It's, what's become really clear here is there are a lot of levers we've got on farm. There's quite a lot of uncertainty. There's a lot of looking at what's going on the ground, test and adjust. And we've got an awful lot we can put together, but it's complex. And there's a lot of things that can adjust and change things. So a lot of it's, I suppose, the art of farming as much as the science. It's looking at integration with our cropping, how we're using the livestock, how we're using the various grazing rotations, things like that, to actually manage the nutrients as they go through the farm. So a lot of what we're doing there, and this was really interesting because it made me sit back and look very much at a number of different tillers that I wouldn't normally be looking at. So that's a real rattle through. Obviously, there's a great deal more on the ground that, at a detailed level, looking at kit and stuff like that we could play around with. But there was a huge amount of lessons learned, or certainly lessons identified, and I hope we've learned at least some of them. Tabitha.
Brilliant. Thanks, Tom. Now, I think, John, you're going to take control of the questions. I think Nigel's been answering some of them, but if we can go through the top ones, whether there's an answer or not, I think.
Yeah, that sounds great. Yeah, keep the questions coming, everyone. First up, perhaps for you, Tom, was there any difference on the fecal counts during the trial between control and Willow group? I know you touched on it in your conversation. Could you perhaps sort of elaborate a bit more? What are you looking at, and any results that you've seen?
The answer is not noticed, but we didn't focus on that probably until too late. My instinct is that because both of the groups were on the same ground, apart from the Willow, I think my feeling was that they were getting quite a lot out of the ground, which is why the actual Willow piece wasn't so obvious. So the answer is no, but we do look at it a lot to see what's going on. And I think generally speaking, and indeed we didn't need to worm them. So they were both getting something from somewhere.
Yeah.
It's worth noting on that, not for the Willow, but in terms of looking at the lay and what you've got, what they're grazing. There is another IF field lab happening in Devon, is that right, Laura? That's been looking at that and is still looking at that sort of thing, that whether obviously there's a nutrition piece there, but also whether the tannins and stuff available in the forage is enough to reduce your need for worming.
There is something on that might be relevant. That particular field, and again, this just to make Nigel's life really difficult.
We would never do
generate
that,
some more
Tom.
generate some more uncertainty. One of the things we have done, it's actually a standard clover lay, which is 4 years old. We have let it flower and seed a couple of times, which of course it makes it more persistent. But the other thing is we have dusted in quite a lot of sort of supportive forbs, especially things like a bird's foot trefoil, sanguisorba, plantain, and a lot of those are known to be anthelmintic. So that might have skewed that bit.
Thank you. And I know Nigel, you've answered a couple, but just for sort of wider benefit of the group, someone asked, do you supplement selenium in the UK? John answer that. Perhaps you could answer it for everyone.
It can be fairly widely supplemented. You've got the slight issue there that Mindrum is organic, so we shouldn't really be prophylactically supplementing without showing need. One of the big problems I had on trial, if we looked at the glutathione peroxidase coming in, there were animals that were in the normal and marginal. So the glutathione peroxidase was a lot better when they were immediately weaned coming in, suggesting the previous fields were higher, and we'd gone into that field that it was a field effect on the one we gone in. So that field had a lower selenium uptake for some reason, whether it was the underlying geology, whether it was the current weather, whether it was the plant species that were there. But we weren't really expecting that. We weren't really necessarily hugely aware of selenium issues on the farm. We were aware that there were cobalt issues on the farm, hence we were there, but we weren't aware of really ongoing widespread selenium issues. So that's why we didn't actually supplement that head on the trial.
Okay, great. And then another question for Tom. Where are you planting your willow? Within hedges, individually? Are you creating pollards? This is from David. Traditional late summer feed source. Yeah.
Great. I'm planting it everywhere, which I know is utterly unhelpful. What I'm doing is we've got quite a lot in we've got quite a lot in our boundaries and our hedgerows. Largely because we've got quite a lot of wet bits. Obviously, willow doesn't necessarily need really wet ground, but it can tolerate it in bits where sometimes others can't. I'm also using quite a lot of the cactus guards and planting it in the middle of fields. We're doing a bit of an experiment at the moment using cactus guards as electric fence bearers. And I'm trying a little bit putting half cactus guards in so that the willow establishes, and by the time it sticks its head above the cactus guard and is browsable, the sheep can get it and manage it. So we're trying to get it all over the place. And we are using a lot of goat willow because that's what we spotted. But I'm also trying a few other willows as well. And I've been bending poor old Nigel's ear on a fairly regular basis. And so we're trying a little bit of diversity in this as well. So the answer is we're putting it where it makes sense in the ground.
Thank you. Yeah. I'm referring now to the comment section. There's someone actually quite interesting. Kevin's talking about the potential for you, Tom, to trial a variety called Endurance, which was shown to reduce. Are you? You're about to.
I'm writing it down now. It's great.
I was about to say, shall I jump in there? Endurance is one of the short rotation coppice varieties around. There are quite a few of those that have played with. I can't remember whether Endurance was actually the top one we got for yields with for cobalt on it, but I've been trying to get. It's a thing. We've got all these varieties. We haven't actually necessarily got the figures of which ones are really good for that. I've been trying to get funding to look at the a lot of these short rotation coppice varieties as well as natural ones to know things like what leaf yield getting, what the back underlying protein and energy is, as well as the mineral composition of the different varieties. We've done the odd little sort of have a look see and do that, and I can remember that there was definitely one of the short rotation coppice varieties that we found was really much higher. I don't think it was actually endurance. I think it was a different variety. But equally, yeah, the endurance is into the I know has been used with the Belfast group on the greenhouse gas reduction. But then again, that's whether the other varieties are also going to reduce the greenhouse gas reduction. That is also something that I have previously had in a unsuccessful grant application to look and going, okay, what's the effect of the different varieties on like the greenhouse gas production? So there's huge amounts of potential here, just we don't necessarily know enough.
I'm going to use that opportunity to segue into, I've got a poll for everybody to answer, so I'm just going to pop it up. This is just to get kind of a bit of feedback from you all, so you just need to click which box suits you best. But this is in terms of what do we look at next. As you can see from what Nigel's just said and from what Tom's been saying, there is many things that we could be looking at, but we would like some direction from you all about which one we actually need to focus on. There is a possibility that we can do a second IF trial if we can make sure that we're kind of going to get the impact that we want. So there is potential for another big trial. There's also some other work that we can have opportunities for, or it might support something in the future. So yeah, please answer the poll and we'll come back to the results. I'll leave it up there for a few minutes and we can probably keep going on if there's any other questions, John. But that if people can answer that while they're listening, that would be very helpful.
Yeah, great. I guess maybe for everyone's benefit, Nigel, I know previously when we've done work, you've actually found there can be a fair bit of variation between sites and in terms of cobalt content within leaves. Perhaps you could talk about that maybe a little bit in terms of what you found and things like that.
There may be site effects, but whether they're truly sites, we had a little bit on some, or whether it is come back. Willow will be, well, most of the mineral uptake will be dependent on soil pH, and Willow especially is very sensitive to the redox potential of soil and basically how wet the soil is. So in a wet year, cobalt uptake is greater. So assuming this goes, it's definitely in grass, assuming that goes to Willow as well. So there's like many factors on that. I wouldn't just like to, there will be the underlying cobalt in the ground will be in effect, but there are so many factors on taking up. Hence sometimes that soil mineral samples don't actually reflect what you'll get within your grazing and hence your animal.
Yeah. And Tom, from your weather data, was it a wet year? Was it a dry year? How did it stack up?
Well, it was interesting. It was slightly wetter. We normally get 750 mils of rain, and we got 820 last year. And that's December to December, sort of calendar year, which I know isn't entirely ideal. But we got the rain at almost exactly the different times of the year. So it was, and I think this is one of the really interesting things. It's this diversity, because of course that means that different growing conditions, different times of the season, we're actually now recording a lot of temperature as well to try and see if we can work out if there's any sort of relationship. But it was and I suppose 70 mils over 750 is 10% difference, but actually the real thing was it was the timing of the rain that was so fascinating. And also, of course, the previous year it was that really cold, hard spring we had. So it's this whole thing going together. I think it's the key lesson for me is very much we've got to be able to deal with diversity.
Yeah, that's interesting. Another question from Jenny. Are you looking to do a trial with your cattle, implementing the lessons that you've learned from this trial potentially? Is there any interest in that, or is it sheep specific at the moment? What are your thoughts?
I'm doing punter research on cattle. Our cattle were moving pretty hard and fast around the farm at the moment. We've got quite a lot of agroforestry, quite a lot of these cactus guards in the cattle fields, providing lots of all the benefits we all understand about that. But I'm not really in a position to do any meaningful stuff I think, in terms of data gathering on that yet, until the volume of the willow in the fields concerned has grown up. I think that one of the tricky things that, at a practical level here, is especially working with 580 lambs, is that's a lot of willow to cut, or to actually have on the side of the piece. So, and certainly putting it in meaningful, and I think the thing that Nigel raised so rightly was that getting the meaningful maths in terms of how much we've got to get into them would be quite tricky at a practical level with us at the moment. I haven't got SRC or anything like that I can deploy into them. So the answer is, we're observing, we're watching what they're eating, and I spend a bit of time sitting on the side of the hill watching them, because that's where you learn. But probably it'll be a couple of years before we're in a position to do anything meaningful from an academic point of view.
Okay.
Right, I think we might need to move on to some next steps. Anybody, we'll have a look at these questions afterwards though, and with the follow-up, we'll answer the questions that we haven't managed to answer. We have been answering some of them either in the chat or in the Q&A as we go, so if anybody wants to have a read, but we will send answers around afterwards as well. I will. So one of the I'll share the slide again, but one of the things that we were talking about in terms of next steps, so that's brilliant. For anybody that hasn't answered the poll, please do answer it. And the slide I'm just about to share, which I'll let John talk through, is actually in terms of doing some mini trials. So we did have some funding for mini trials last year, but due to various reasons, we didn't actually manage to get them going. So we have still got potentially some money there if people want to have a go without it being part of the kind of, it'll feed into this project that we've done basically. So John, can you explain a little bit about what that's with and how that's working?
Yeah, so the hope was maybe to have 4 or 5 farms doing this, what Tom and Nigel have been doing on his farm, hopefully spread around the UK, so we can understand that sort of geographical element as well, how it might work for different farmers around the place. Again, incorporating different sheep breeds, different systems, that sort of element. The funding's still there. We have, for lots of reasons, didn't quite manage to get it across the line, but so I guess call to action, anyone that's on the call that has flock and an interest in this, please seek us out, and here's the suggested protocol that we sort of came up with at the time, and I know everything's all up for discussion. It's a fine balance between that sort of trying to produce something scientifically or half scientifically rigorous, and at the same time making it work for farmers in their very busy lives and days and things like that as well. So very much interested. Yes, seek us out if it's something that you think you'd be interested in hosting on your farm going forward potentially.
So with that, I'll pop up my second poll. I think I might have to end oh no, I think I can put them both at the same time. No, might have to end the first one. 1 second. So this next one that I'm going to pop up, which again I would love you to all answer, is about being involved in the future. So this, you're not being committed to anything. We're not collecting any data apart from numbers on this, but it's just to get an idea of whether you would like to be part of the whole field lab, whether you would like to do a mini trial. So there is this protocol that's on the screen that obviously involves a fair bit of activity. It's still less than Tom had to do, but still quite a lot. If you would like to be involved in a mini trial, but just do not think that this is feasible, that's on the screen, there is a box for that. It's the third box. It's got with adjustments on there. So please tick that one instead. If this has been a complete overload on information and you are done with Willow and lambs, then there is a box for that as well. But we just wanted to, this is just a bit of a gauge of interest. There'll be an opportunity for a proper expression of interest coming round with the follow-up email and stuff. But it was just so we could get a bit of an idea of whether we need to review this protocol or whether it's going to be feasible. And I think we can probably, where are we on time? We've got 8 minutes left. Shall we pop on to, any more major questions, John, or should we pop on to the upcoming events?
Yeah, should we pop on to the upcoming events? If we've got a bit of time, I could do maybe 1 or 2 questions at the end, potentially. So first things first, worth saying that these slides will come round to you, and they're all linked. All these events are linked. So please click on the links and things like that. But this page in the top right hand corner, Soil Association events page, there's loads going on up there. I guess what we've got listed here are the tree and sheep themed events, thinking that that's our audience, but there's definitely a lot more going on, so please refer back to that web page where there's all sorts happening. So for me, the next major event is a practical farm event, Brooksbury College Hall Farm, so that's the 13th of March. Very much sort of looking at how they've integrated trees into their farming system. It's a mixed farming system, a bit of a farm walk, and then they'll talk about their experiences of BNG and how they've used SFI and things like that for how they've integrated trees. So really good, exciting farm walk, I think, and that's in Leicestershire if people are local. And then before I hand over to Tabitha, so we've been running a whole farm tree plan project, which has been funded by the Forestry Commission. It's a project to look at supporting farmers to understand their existing treescape on their farm. We're sort of calling treescape, but meaning sort of hedgerows, woodland, maybe infield trees, things like that, everything. How with a few tweaks to management and things like that, their treescape might better support their farming business. So we've been running sort of 30 pilot farms around the UK around England looking at this. And this is the end of project webinar on the 30th of April. We finished March, which feels quite daunting because it feels like we've still got lots of work to do on that. But yeah, Tabitha, you want to talk about the sustainable sheep production?
Yes,
events?
So when you get the slides, this isn't actually a link yet because I haven't got them all set up yet, but we have teamed up with the National Sheep Association this year to deliver at a lot of their events and stuff, some work that we've it's where we're focusing a bit for one of the projects. So it's a lot of sustainable sheep stuff. So we're going to be doing a webinar on, I think, the 15th of May, kind of introducing it. So it's going to be about soils, grazing strategies and schemes. And then we're going to do a more practical version. We've got a seminar at Welsh Sheep, and then we've actually got a stand at North Sheep. And we'll be doing a workshop looking at soils and stuff, and talking through all things schemes, whatever the updates are at the time, at that as well. So please do, if you're going to be at the NSA events, do come and see us. That would be lovely to see you, and look out for the link for that webinar coming around as well.
I'd probably be sacked if I didn't mention the Agroforestry show. So this is the second edition. It's happening sort of every 2 years. 10th and 11th September at Wood Oaks Farm, which is a Soil Association land trust farm down in Hertfordshire this year. We tend to try and move it around. We are trying to get it further north each year, but it's the sort of challenge that we face. Again, a great show how we can integrate trees into the farming system. We're sort of building on last time's successes. So we had about 1,300 people there last time. Hopefully we'll get a few more this time, build on sort of the practical demonstrations. So it is incorporating a little bit of short rotation coppice that I think a bit is being planted now there as well, looking at sort of sheep connection, but also, farm walks, lots of talks talks and things like that. We're about to open the second session proposal. So if anyone on the call has any sessions or anything they'd really like to speak about, then please get in touch. And then finally, so the next Willow for Livestock webinar, this network that's running this talk, date and topic not yet confirmed, but please a call for people to reach out to me with topic ideas and questions. You know, perhaps you're trialing something on your farm, or perhaps you have something to share or anything. Please reach out to me, and we can get a date and a topic in the sort of, you know, in for the next event. I think that's it. How are we doing for time? 2 minutes.
2 minutes. If anybody hasn't answered the poll yet about kind of whether you want to be involved in anything, please do. But again, like I said, the proper expressions of interest will be coming with the follow-up email. But it's just for us to get an idea of where we should be, what we need to do.
Final question: did the study last up to slaughter? If so, is there any difference between the two groups? From Aubrey, who wants to take that one? Nigel, Tom?
We have been, we were drawing lambs from about halfway through. I don't think we've done a full analysis on the culls. We've certainly been recording.
I did a quick add up, and it was, I think actually we had slightly more we had like, it was 1 or 2. There was no significant difference, but there was 1 or 2 more from the control group went to slaughter than from the test group.
They were going in lumps. Sometimes there'd be a lump of Willow, sometimes a lump of control. It was fairly random. I don't think, looking at the kills coming back, there was any major difference on the kill out sheets or anything on that, is it either?
No, well, that's what Sam's reward was for, was how consistently well that all the lambs scored or graded. So there wasn't anything obvious in terms of when there was a lot of Willow lambs that they were grading better or anything, because everything was very consistent on the grading sheets, weren't they, Tom? So
yeah, that's right.
Okay. We got a few more. There are a few more questions and things, and we'll go through the Q&A afterwards and make sure we try and answer any that have fallen through the net. But thank you very much for your questions, and thank you for coming today. Thank you obviously for Tom, Nigel, and Tabitha talking about their trial and the progress, and good luck in everything, you know, going forward. There will be a follow-up email with slides and more information and things like that as well. Please refer to please refer to the Innovative Farmers website, where we've got recordings and things like that from previous sessions. So we've talked about sort of propagating and lots of different sort of other Willow themed talks. So please refer back there if that might be of interest to you. But as I say, thank you very much to everyone that's contributed, and have a good day. Thank you.
Thank you.